Good evening, everyone. Welcome to Botany Insider's new free previous year practice session. Let's wait for the students to join in and then we'll start off with our class. Okay. Just confirm whether the audio and the video is clear to everyone or not. Those of you who are there in the class right now, just confirm whether the audio and the video is clear. So in this particular class, we'll be discussing or we'll be starting with the part C solution. Okay, till now we have done a number of questions related to part B. From this particular class, we'll be discussing the questions of part C. Okay, so let's begin with our class now. If you're new to Botany Insiders free sessions, if you're new to our platform, do not forget to hit the subscribe button. In addition, we have our own application. You can download the application from the iOS as well as from the Play Store. Okay, it's available for both the Android users and the iOS users. Then if you are sincerely preparing for the December examination, so we have two courses running for the time being. First one is your full course and the second one is a crash course. If you wish to enroll in any of these, you can download the application and get yourself enrolled. In addition to that, you can also place your orders for your notes hard copies specifically curated for the December examination on like either via the WhatsApp or via what sending SMS on this particular number. Okay, via your WhatsApp number, you can place your orders or via the application, right? Let's begin with our class now. And in addition to the same, we are about to start a previous year dedicated course for the upcoming examination. It will be a revision plus a PYQ course. Okay. So we are about to start with the enrollments of the same. Download the app to like get your slots booked at the early. Let's begin with the class. This is the very first question, everyone. All the questions that we are going to discuss, they are of part C, morning shift. Okay. Part C, morning shift, June 2023. Write down, like, this is the very first question for you all. The question is, the following statements are made for the structure of a 30 nanometer chromatin fiber. You need to find out which of the following combinations of the statements are correct. Okay, read the option carefully and then mark the answer. This question is from nucleosome concept. Okay, this is from nucleosome concept, unit number 2, that is cell biology. Try to find the answer. It was a straightforward direct question. Mark the answer for this question, everyone. Take your time, understand what the question is asking you, and then try to mark the answers. Use elimination technique, use your basic framed knowledge, and then try to eliminate the options, try to eliminate the combinations as and when you get sure about a specific option to be correct or incorrect. Okay, give it a try, then we'll solve out the question, everyone. Very simple question. If you know about the the complete concept of this nucleosome, you will be able to mark the answer easily. Okay, solve the answer for this and mark the answer in the chat section. Take your time, mark the answers, everyone. Give it a try, everyone. Simple question. Very good, very good. Everyone, mark the answers. Very good, Anjali. Very good, Nisha. 15 of you are there in the class right now. Only 3 have marked the answer. Even if you get the answer incorrect, that's absolutely fine. But trying out the question at the first go is important. Okay. Very good. Okay. So let's look into the first statement. First one is like, see, when we look into the nucleosome concept, in this, what do we study? We study that how does the chromatin fiber gets arranged in a compact manner so that the entire structure can be like concised into a chromosome, right? So for that, we majorly have two of the models. First one is the zigzag model. Second one is a solenoid model. Okay. Solenoid model is the one which says that you have a central line, or it considers a central line and around which we have the coil getting done. Okay. Of the chromatin fiber. Zigzag model says about, or it talks about the formation, or it talks about the organization in a zigzag orientation. Let's see. In the solenoid model, the linker DNA connects the consecutive core particles. Okay. Second option, in the zigzag model, alternative chromosomes become the interacting neighbors. Okay. Third statement, in the solenoid model, 12 nucleosomes are arranged into separate stacks, whereas 8 nucleosomes per turn make a single stack in the zigzag model. Okay. Fourth, H1 histone is essentially required as per the zigzag model, but not in the solenoid model. If you look into the fourth option, this one is for sure incorrect because H1 is a linker histone. It is majorly required in your solenoid model. So D gets incorrect. If I am sure about D being incorrect, I can eliminate option number 1 easily, right? Then you have the chromatin fibers prepared with H4 histone that lack their tail could fold into higher order fibers. It's talking about the H4 histone, and it's talking about the tail. When did we study the H4 histone having the tail? Which particular histone have the tail? Yes. Which particular histone have tail? Write down in the chat section, everyone. There are in total two types of histone, core and the linker one. Linker is H1 and the remaining four, H2, H2A, H2B, H3 and H4. Out of these five, which one is having the tail? Yes. Think about it, everyone. Write down in the chat section which particular histone have a tail. H1 histone, right? It's the H1 which has the tail. So E option gets incorrect. So without understanding or without having the knowledge of any other option, I can eliminate D and E very easily, right? And if I eliminate D and E, I can eliminate three of the combinations which are given to me, and I can reach to my correct answer as A and B only. So the correct answer for this particular question is A and B. First and second statements are correct. Third statement is saying in the solenoid model, you have two separate stacks getting formed and then 8 nucleosome per turn, single stack, all these things. We did not study about the nucleosome arranging into two separate stack in case of your solenoid model ever. So C option is also incorrect. Okay. I am expecting this question is clear. Understanding how to solve a question, that is of utmost priority. Okay. And for the same practicing, for the same learning, we are starting off with a course which will help you out in understanding the concept, which will help you in understanding and making sure that you know how to mark down the answers. Okay. This was the first question. Now, this is the second question for you all. This one is related to unit number 13, that is methods in biology. Okay. Now, you have to mark the answer for this particular question. Easy question. You're given various techniques and their main characteristic features. You need to find out which of the following is the correct one. Okay. You have to find which of the following is correct. Very simple. If you can recall what Khushi ma'am taught you in the very first class of hers related to unit number 13, methods in biology. If you remember that, you can easily solve out the answer for this. It's very, very simple. Okay. In addition to that, related to this RACE, that is race, which is one of the technique in related to this, like this thing has been asked 4 to 5 times in the same format, in the same match the following format. CSIR has asked the question where they have taken RACE as one of the options. Okay, try marking down the answer for this question, everyone. Very simple question. Think about it. If you can eliminate it with the help of your basic understanding, you can mark down the answer easily. Everyone participate. 13 of you are there in the class right now. Mark down the answer. Okay. Let's see. First option is like I remember about RACE. That is, it helps in determining your ends. Okay. This is something that I can recall. Okay. It helps in determining the ends of the same. That is race cloning of the cDNA's end. All I remember is that RACE is somewhere related to ends. Okay, so the cloning of the complementary DNA ends. So if I mark down D with the third option without even reading any other option, I can reach to my correct answer because answer is option number 1 itself. Okay, all of you got the answer incorrect. Remember about this particular thing. Okay, race is related to the cloning of your cDNA's end. This is the most, like, so many times this particular thing has been repeated. Okay. Then you have first option, that is reverse transcription. So cDNA library, complementary DNA library. Then you have southwestern blotting. This is used for the detection of the clones, encoding the nucleic acid binding proteins. And then you have the genome walking PCR. So this is related to your second option, that is the determination of the 5' and 3' sequences flanking in the 5' known region. Okay. Very, very simple, easy, straightforward question. Okay. First is the correct answer for this particular question. I hope this is correct. This is clear to all. Next question is related to your cytoskeleton. Okay, this is related to your cytoskeleton. Again, a match the following type of question. Give it a try. You, on one hand, like now in this question, you are given 3 of the columns. First column is microtubule actin. Second column is you are given few of the proteins. And in the third column, you are given few of the functions related to the same. With the information that you have, try to find out the answer. Whatever information that you have, try finding out the answer for the same. And it's easy. You can mark down the answer easily if you read it properly. Okay. If you understand it properly, you'll be able to mark it. Take your time. Go through this question, everyone. Very simple. Try out everyone. Very good, very good. It's okay. Nisha, even if you're not sure about it, it's okay. We'll give it a try. Okay, we'll understand the main thing, but try to at least mark the answers. Okay, that's very important. Done, everyone? Mark it quickly. Okay, let's look into the answer. The answer for this particular question is option number 3 itself. Very good, Nisha. Answer is option number 3. Now, see over here. Microtubule. Microtubule is related to what? Microtubule is related to your, like both of these are related to your cytoskeleton itself. First is the microtubule, second is the actin. So microtubule, third and the first one, that is in the microtubule, it helps in the filament cross linking. Second, it is over here we have a number of filaments present. Okay, in addition to that, X, 1 and D, that is 1 and D. So first, like for your microtubule, first and fourth statements are correct. In addition to that, you have tau and your catenin working in. For the second thing, that is your actin. For the actin, there are two main things, that is trophomodulin and glycin. In addition to that, you have filaments and they bind to the plus end, and then you have the minus end as well. Now you have to correlate it to the assembly and the disassembly which occurs via your treadmilling concept. We have discussed, like for the students who are there in the course, we have discussed about the actin treadmilling, right? In the actin treadmilling, what did we understand? We understood about the G actin, and then you have the F actin. G actin, which is in the active format, gets binded to the plus, and then there is disassembly on the minus end, etc., etc. If you can recall the thing, so I can easily correlate actin with the plus end and the minus end. So if you observe in the entire thing, Y with B, like Y with your B and C is there only in option number second and third. So first thing, you can filter out your options with the actin thing, and second thing, you can filter out with the help of your microtubule and your catenin as the protein. We have discussed about the microtubule dyne, sorry, that is a different one, but still catenin and your tau is the protein which is related to your microtubule. Okay, remember about this particular thing, and with this question you can take up the important information that is there in this question. Okay, try to keep a note of this question, because you might get the same question or you might get some information from the question repeated in the next one. Okay, I hope it's clear. One more question for you all. This one is from your ecology unit, or ecology or evolution combined, because this topic is like a common topic for both the units. It is related to the theory of kin selection. You have to find out the correct option. Okay. Theory of kin selection. First option, second, third, and fourth. Four options are there. Give it a try to this question, everyone. Uh, if you know what is kin selection, if you know what altruism is, you can mark the answer easily. Give it a try, everyone. Solve out, everyone. Read what is given to you. Understand what is direct selection, what is indirect fit, what is direct fitness, what is indirect fitness, etc., etc. Anjali, read the question again, understand the question. Nisha, you also read the question carefully. What is asked and what are we correlating it to? Try it out, everyone. Kin selection, theory of kin selection. There is no E option. Yes, done everyone. Very simple question, right? We are going to discuss each and every question, like whatever is given on the screen. We'll be discussing every question in detail in our main PYQ session, which is about to start. Like the enrollment will start on Saturday and the main course will start on 18th. Very good, very good. Answer for this is option number 3. Those of you who have given the answer as option number 4, just try to recall what altruism is. What is altruism? Altruism is a like behavior in which the same species member, by taking risk at their own life, they try to provide help to their own well-being, to their own species members. Why? So that they can provide direct or indirect fitness. So a gene of altruism will spread in the population. When will it spread in the population? It will spread in the population if via this act of altruism there is increase in the fitness. That is the main concept of kin selection, right? That is, you are, yes, it is selfless behavior. We, like, if it is maintained at a very low frequency, then the work will not be done, right? And altruistic behavior and gene level is not very much correlated, right? It is your fitness which is like more interlinked together. So you have to look into the fitness concept. So first and second option, only direct, only indirect can be eliminated. Fourth option, they are talking about the low frequency and all those things. This also gets eliminated. You can easily reach to the answer as option number 3. Are you understanding this? It's a simple question. You just have to understand what the examiner is asking you. It is asking you the correct option in terms or in accordance with your kin selection. Are you understanding this portion, everyone? Yes or no? Is it clear to all? Yes, let's move forward to the next one. Okay, leave this. You can do this particular question now. Very simple. This is related to your meta population dynamics and habitat conversion. Very simple question. Think what the question is asking. Okay, do not be in a hurry of marking down the answers. Think what the question is asking you. You have to find out all the correct options. Lena, don't be in a hurry to mark down the answers. Read it properly. Even if it is correct, even if it is incorrect, doesn't matter. But take your time to read the question at least twice. Take this thing in habit, everyone, that you are at least reading question twice before marking the answer in your main examination as well. Mark down the answers. Correlate what is getting asked and think what do you mean by meta population dynamics and habitat conversion. And remember one more thing, that in the examination you will not find every particular point read by you. Okay, there will be new surprises, there will be new information that would be there in the examination in most of the questions. So you should know the trick, you should know the strategy of solving out such questions. Okay, like in this question, option number B is something very new, it is something which is very different or a new information for you all, right? So do not fear from this new information, try to find out the answer according to your understanding of the concept. Okay, one more minute, then we'll start with the discussion of the same. Understood? Let's look into the answer now. The answer for this question is option number one. Very good, Isha, Nisha, and Anjali. Answer for this is option number one. A and C is the correct one. Look into the first one. The size of the suitable patches are important because the demographic statistically can lead to the extinction, specifically in the organism with low reproductive output. It is saying the size in which the meta population is uh, moving from one location to the other one. What is meta population dynamics? We have discussed about this thing in detail. Meta population dynamics is that from the entire patch of the population, some of the individual decides to move to the other location with like very dynamically without any proper reason for it. They keep on migrating from one location to the other one. Some might come back to the same one, some might go to the other location. So it is saying the size matters, because if their reproductive output is less, they might get extinct. A option is correct for sure. If A gets correct, I can eliminate second and fourth, because these are the only two options. Without even thinking of any other thing, I need not to even read option number B, because in the examination I have to save my time, right? So I will not be reading option number B during my examination time. But when you're preparing, when you're actually like practicing out the things, you should read all the options and take out information and take out each and every possible content from the questions, because that's also important. Okay, then you have option number 3. From the conservation perspective, large number of suitable patches are not sufficient if the distances are too large, preventing recolonization and the rescue effect. That is also very correct. It is saying that if by chance you have the population moving to some location where the size is bigger, but the distance between the two areas is very large, at that time, no matter what the distance is, no matter how much, sorry, no matter how large this particular area is, you will still, like, there will still be prevention of recolonization. Recolonization means this organism group coming back to the parental group. So this is also a correct statement. Then, to minimize the extinction risk, there should be a low variance in the local patch quality as much as possible to allow for synchronous dynamics. It is saying in order to reduce the extinction rate, in order to reduce the impact of a species like getting completely extinct from an area, there should be lesser variance in the quality so that dynamics can be maintained. This option is incorrect. This option is completely wrong. So I can reach to my correct answer with option number 1 and 3. So with option number A and C being correct, so the combination of A and C, which is your first option, is the correct answer. Okay, so these are the questions for today. I hope these are clear. We have taken up 5 questions. I hope these are clear. And if you want to have in the similar pattern and in a much more detailed pattern discussion of the questions, make sure that you are like joining in on our application. And also we are about to start with a new course. You can download the app, and like from day after tomorrow we'll start the enrollments for the same, so you can download the app and uh, get your early bird 10% off offer as well, right? In addition, for the time being, we have 2 courses running right now, and 1 hard copy notes availability. So any one of you who wish to enroll, you can download the application and get yourself enrolled in the same. Okay, this was about it. Let's meet in the next class, everyone. All the best, good luck, take care, bye everyone. Yes, part A session will also be there in the course. In this PYQ course that we are about to start, we'll be having part A sessions as well. Okay. So in the PYQ session, you'll see part A, that is your general aptitude, also getting discussed uh, year wise. Okay. And in the courses also that are running right now, we have part A also available. Live sessions are there. Okay. I hope it's clear. Shaik, you can WhatsApp me on this number. You can have a discussion with me. Okay. Bye, everyone.